Okay, so we are going to move to the next session. And the next session will be dedicated to resistance and environment and resistance. So, are we online with Paul Verweij? Hi, Paul. Hi, Paul. Oh, yeah, I have to look at the camera here. Hi, Paul. Do you hear us? No, it doesn't work. It doesn't work. Yeah. Hi,
Hello?
Paul. Do you hear us?
Yes, and now I hear you.
Okay, perfect. Thank you very much, Paul, to be with us this afternoon. Until now, we had a very brilliant seminar, and I'm sure we will continue. So, Maiken, if you want, you have a seat here. Okay. So, Paul, you are going to talk on Aspergillus resistance to antifungals. Of course, it is your favorite topic. You have 25 or 30 minutes. Thank you very much.
Thank you, Jean-Pierre. Hello to everybody in Rennes. I hope you're having a good time there. So I want to talk a bit about resistance in Aspergillus, and I will talk about mainly insight we have obtained in the Netherlands, also to avoid overlap with other speakers. So that were my conflicts of interest. So as you know, Aspergillus has a habitat in the environment and really likes to grow on dead plant material. A recent review showed the various compartments where you can find resistance, and Aspergillus fumigatus is in the environmental environment where there's a One Health issue with resistance selection. So as you also know, the azoles are a really important treatment option for patients with Aspergillus diseases, and especially invasive aspergillosis, where we have voriconazole and isovuconazole as primary treatment options, amphotericin B as alternative, and posaconazole as prophylaxis, and recently that was also shown to be good as a primary treatment option. So if you think about resistance, this is a stress response adaptation to stress by the fungus, where you have a population of cells. And when you change the environment, then the cells may become unhappy and unable to grow. But if there is some variation in this population, then there might be certain individuals within the population which actually can grow in that environment and then become dominant. So we have the stress factor, which could be the antifungal. You have the genetic variation, for which the population size is very important. And then you end up with the selection of those best suited to grow in that environment. So this can happen to any antifungal drug, and probably some factors are important if the drug is fungicidal or fungistatic. Also, if the resistance mechanism is simple or more complex, if the exposure is short or during a long term, and also the population size. And I think we could rank the antifungals according to the risk of resistance selection. So flucytosine is always mentioned as the one with a high risk for resistance selection. Then you have the azoles, the echinocandins, and probably the polyenes have the lowest risk. So with Aspergillus, I think the main issue is related to Aspergillus fumigatus and with the azoles, where we have human exposure and environmental hotspots, and these together bring forward the problem of resistance selection. So which clues directed us towards an environmental route? Well, first, the finding of azole-resistant infection in patients who had never been treated with azoles, and also the presence of a dominant resistance mechanisms, which would be unlikely if you had resistance selection in individual patients, and that was the 10-repeat 34L98H. and also the finding of the same resistance mutation in environmental sources. And this is an old picture of our hospital, and you can see a flower bed close to the main entrance where we recovered this 10-repeat 34L98H mutation. The other important factor is, of course, do the fungicides which are used in the environment and are considered the selection factor, do they have activity against Aspergillus fumigatus, which is not the target for the use of fungicides. And indeed, a significant number of fungicides showed activity against Aspergillus fumigatus, and this mutation confers resistance to both fungicides and medical azoles. So this leads to the concept of two resistance selection routes, the one where this resistance is selected in the patient, and the other through the environmental exposure, which we refer to as dual use of mode of action. And it's important to realize that the patient route affects actually just one person, that is the patient, because the infection does not spread from this patient, but that the environmental route leads to a global distribution. And there are many maps which have been published looking at the distribution of azole resistance in Aspergillus fumigatus. This is a map which is now, I think, 4 years old. But on the right, I just looked in the literature, looking at the frequency of resistance in various publications. And you can see in Europe, numerous countries publishing resistance rates, also in the Americas and also in Asia. And the rates, they vary between 0 and 10%. So it's clear that voriconazole or azole resistance has impact on the mortality. And if you look at the early studies, which were published until 2011, there were very high mortality rates between 80 and 100%. One of the problems of these studies was, of course, that we did not have a control group, and therefore a study was performed looking at the difference in survival in patients with azole-susceptible and azole-resistant infection. And the excess mortality was about 20% after 6 weeks. So if you look at those studies and other studies, you can see that there is a significant difference in survival, but in those years, the mortality rate was already lower than in early years. So how can we explain the difference? Well, I think in the earlier years, we didn't consider resistance as a cause of failure, and patients were changed when they failed clinically. But in the second period, already resistance testing was performed and treatment was changed on the basis of resistance testing. So given the relatively high rate of resistance in the Netherlands, we decided to change the guideline in 2017, where we recommend combination antifungal therapy in patients suspected of invasive aspergillosis. And we hope, but that has not been demonstrated yet, that reduces the excess mortality significantly. So when we look at the resistant frequency in our country, we have a surveillance network where we screen for resistance, and we have university medical centers where the resistant frequency is around 11%, and the teaching hospitals which have a resistance frequency about half of that. So over the years, we see that the resistance frequency is variable, but has been above 10% since 2015. And 10% is considered a percentage which when you should consider changing your primary treatment. One of the problems is that this is based on pathogen surveillance. And actually, of course, we want to know what the impact is for disease. So really, we need disease surveillance. But that is much more difficult to determine. So when we look at the our surveillance, one thing which we noted is actually that we see quite a number of resistant isolates which have this characteristic signature of the 10-repeat 34 or 10-repeat 46, but they have additional mutations in the CYP51A gene, or they have variation in the length of the 10-repeat. And this was something which was also observed by Jenny Stelton in the United Kingdom when she did a citizen science project. where people were asked to culture or send samples from soil from the gardens, and they were analyzed for the presence of resistant Aspergillus. And here you can see the number of variants she found in the 10-repeat 34 L98H background. So she found isolates with only 10-repeat with the characteristic L98H, but also a lot of isolates with additional variants. So we were interested to see if that was also the case in clinical isolates. And we've been collecting clinical isolates now for 28 years in the Netherlands. And Yinggai Song, a PhD student from China, she analyzed all those isolates, nearly 12,000 isolates, and also looked at the resistant isolates which had mutations in the CYP51A gene. So that was over 1,800 isolates. And you can see that also in these clinical isolates, in the TR34 background, in the TR46 background, and also isolates with individual SNPs, you can see that there appears to be an increase in the number of variants over time. So overall, she found 34 different genotypes and this apparent increase over time. But we have to be cautious with this because, of course, over the 28 years, the awareness increased and also the number of isolates which were sent to us for analysis also increased. So is this a problem, this emerging of these variants? Well, if you look at the phenotypes, then in the top row, you have the 10-repeat 34L98H isolates in blue, and you can see that most of these isolates have very high MICs for itraconazole. For voriconazole, it's more variable between 0.5 and 32, but most of them have 4 or 8. Posaconazole is more closer to the wild type with 0.5 to 1, and isovuconazole is more similar to voriconazole. And in the bottom row, you can see the 10-repeat 46 group. Most of them have a variable MIC to itraconazole, but they all have a very high MIC for voriconazole. Interestingly, you can see that in the itraconazole group, it seems that there are two populations, one which have very high MICs, and also a group of isolates which have MICs around 1 or 2 milligram per liter. So to determine if these variants have an impact, we decided to determine a core MIC distribution, which is the mean MIC plus or minus 3 standard deviations. And it turned out that 5 of 7 variants were outside the core MIC distribution for the TR34 group, and 2 of 7 were outside the MIC distribution for the TR46 group. So this is an overview of these variants and how this impacted on their phenotype. So this is a group of isolates, which in addition to the 10-repeat 34L98H has 2 additional mutations at position 297 and 495. And you can see that all these isolates have high MICs for isovuconazole. So that's thought to be due to the 495 SNP. But you can also see that within this group, there is some variation if you look at the voriconazole MICs, that some have lower MICs than expected, and some have higher MICs. And this might point towards other mutations outside the CYP51A gene, which have accumulated over time. So this is another variant where there's a mutation actually in the 10-repeat region of the CYP51A gene. And you can see that these isolates have a relatively low MIC for itraconazole. So for the sensu stricto group, it's 27, and you can see that for most isolates, it's around 2. But you can also see that one isolate still has an MIC of greater than 16. One of the problems is that this is not the whole group. There were actually 32 isolates with this SNP in the 10-repeat, and only 7 of those were outside the core MIC. So I think this illustrates that we have to really be careful in interpreting what the role is of these SNPs in the resistance phenotype. And finally, there were isolates which had the 10-repeat 34L98H with 2 or 3 additional mutations, the 289A and the G448S mutation. And you can see that with these isolates, most of them had a low MIC for itraconazole, which is not very characteristic for this TR34 group. and has high MICs for voriconazole and isovuconazole. So I think this illustrates that this is becoming very complex, because if we look in more detail at this mutation, the 10-repeat-34L98H has a high MIC for itraconazole, and the T89A is a mutation which we know from the TR46 background, which, as I showed, has a high MIC for voriconazole. The mutation at 364 is one which is seen in agricultural fungi, but we don't know what the phenotype is in Aspergillus fumigatus. But the 448 mutation is also found as a single mutation and then has high MICs for itra and vori. But if we look at the overall phenotype, you can see that itraconazole is relatively low. Difficult, because the last variant might have 3 markers positive, and then you don't know if it's a co-infection of 2 resistant strains, or might this be the new variant. And with increasing new variants, it will become more and more difficult to predict the exact phenotype. So how to solve that would be to actually analyze and do gene editing studies to see what the contribution is of each of these SNPs to the resistance phenotype. And that is a lot of work if you have to look at all these different variants. So when we look at the environment, of course, there are several hotspots which have been identified, including flower bulb waste, green waste, wood chipping waste, and strawberry waste. And the number of hotspots which have been described is increasing. So the hotspot is characterized by the presence of Aspergillus fumigatus and also of azole residues. So the question is, because there's a lot of flower bulb production in the Netherlands, if that is an important hotspot for infections in patients. So if you look at where the flower bulb industry is located in the Netherlands, it's mainly in the northwest part of the country. When we then look at our clinical surveillance, you can see that 2 hospitals, the closest to this region, actually have the lowest resistance rates, while other centers, such as in the north in Groningen, have higher resistance rates. So that seems not to match very well. Also, if you look at the distribution of resistance mutations in the flower bulb waste, it's more dominant the TR46, but in patients we see more dominantly the TR34. So this remains an important question. What is the contribution of a hotspot to actually clinical disease in patients, and what are the transmission routes? So as you will have heard from Joanna Rhodes, high-resolution typing through whole genome sequence is very important and useful to track and trace and to see if we can get more insight in these transmission routes. So she will have shown her work she has done with Aspergillus fumigatus, describing these 2 clades where resistance is dominantly found in clade A. Also, they showed that the clinical and environmental isolates overlap if you look at their sequences, and also that there are some isolates which are quite closely related, originating from the environment and from the clinic, indicating that there is a relation between these 2. So together with Evelien Snelders from the Wageningen University and her team, we decided to look in more detail and take isolates which were from hotspots. So there were 3 flower bulb farms in the northwest of the Netherlands. And we took 85 isolates from that part. And then we decided to look for clinical isolates in Amsterdam, for which we thought that the patients would come from the region of the northwest of the Netherlands. And also patients from our hospital in Nijmegen, which is to the east. So we sequenced these isolates, and then we looked if there were matches between the environment and patients. And we found that there were 4 genomic matches between these 2 sites. So I show one of these matches with 3 patients which were administered to our hospital in Nijmegen. So the isolate from the environment was cultured in July 2016. And we found a patient who had an identical sequence in our hospital in January of 2016. So it was a patient who had a stem cell transplant, had a diagnosis of probable invasive aspergillosis, was treated with voriconazole, but during treatment had a positive sputum sample with a resistant isolate. This was the typed isolate. At the same time, an increase in serum galactose-9. So this indicated failure to treatment, and this patient responded to micafungin. The second patient was found 2 years later in 2018, also with a 10-repeat 34L98 mutation. So this patient was actually from a different department, also had a probable invasive aspergillosis, had a BAL culture positive with a wild-type isolate, but during treatment had an azole-resistant isolate in sputum. And this patient was treated with liposomal amphotericin B and responded to that. Later that year, in June, there was a third patient, and this patient was diagnosed with influenza-associated pulmonary aspergillosis. So it was interesting that this patient did not originate from our hospital, but actually from the south of the Netherlands, from Heerlen, and she was transferred to our ICU. And on day of transfer, we did a bronchoscopy with BEL, and then we cultured the resistant isolate, and this patient died despite treatment with voriconazole and liposomal amphotericin B. So if you look at the matches, you can see that there were matches between the hotspot and Amsterdam and then the patient from the south of the Netherlands and also from our hospital. So I think this is the first time that there's a direct link between a hotspot and the azole-resistant invasive aspergillosis cases. What I found concerning is that if you consider the number of CFUs which are present in the hotspot, which is very high, and you take only 85 isolates from that, you already find 4 matches in hospitals. The other concerning part is that the matches were up to 34 months after the source sampling.
So, no more online connection. Okay, I think he was near to the end of his talk. Do you think we move to the next one?
Spores are released from the hotspot and then travel through the country to the patients. So this is possible because western wind is the dominant wind in the Netherlands, and this could be a way how the spores are transmitted. Another possibility is that there is contamination outside the agricultural arena, and that there are other places in the country where you have selection through the presence of fungicides. And that would mean that there's also levels of fungicides in home environments and in other environments in the country. And I found one study which was interesting. This was also a citizen science project where they asked people to send hair to a central lab, and they looked for residues of pesticides in the hair of these people. So they analyzed 300 people who sent back their hairs, and you can see here the frequency of pesticide detection. So in the Netherlands, right on the right, it was 28% of people who sent in their hair sample. And in Belgium, it was 45%, and in Poland, it was 47%. So quite high percentages of residues of pesticides in these hair samples. They looked for 30 different pesticides and detected 25 different ones. And on the right, you can see tebuconazole, which is of course of concern if we think of resistant selection in Aspergillus fumigatus. Also, it turned out that people who worked in the agricultural environments, they had a high frequency of detection of these fungicides. But also people who live in urban areas still were positive in 1 of 5. So this might indicate that even in our direct environments, there is pressure of azoles, and this might contribute to the transmission from a hotspot to the patient. So I think that remains an open question. What is the dominant route of resistance, and how do patients get infected? Is it through the agricultural route, or could it be through other routes, for instance, through preparation of food at homes? So there was also an interesting study from France where they looked at this question, and they looked at the clinical isolate from a patient who had a 10 repeat 46 infection, and they found isogenic isolates in the home of the patient and in the garden of the patient, but not in surrounding agricultural fields, underscoring the need, I think, to explore in more detail how this home environment contributes to the risk of getting resistant isolates. So have we learned from this dual use of azoles? Well, I want to close with a new concern where we have a new fungicide, iflufenoquin, which has been authorized for use in the United States and in Australia. So this is identified as a DHODH inhibitor and has a medium to high risk of resistant selection. So as you will know, Olorofim is also developed for clinical medicine and is active against azole-resistant Aspergillus infection. So there is now concern, of course, that we have a new azole-resistant scenario where selection occurs in the environment, and Mike Bromley and his team in Manchester indeed were able to select for Olorofim resistance through exposure of the isolate to iflufenoquin. So I think this is a concern for us for the risk of resistance developing against this new mode of action. So in summary, I think that the emerging of resistance variants is something which may present the next challenge in azole-resistant Aspergillus fumigatus, that we still do not understand the transmission routes very well, and that will require, I think, further study. I think another question which we have to address as community is dual use. Can that be safely be used when we introduce new mode of actions in the environment and in medicine? So that's it, and I thank you for your attention.
So thank you very much, Paul, for this very nice talk, many data. Do you have some question? Yeah.
Is there any way to have some kind of regulatory involvement forbidding certain use of drugs in prevention of these diseases in agriculture?
Yes, thank you, Esther. That's of course a very good question. I don't think that we want this to happen, but you know, the problem is that looking at activity against human fungal pathogens is not part of the assessment of these new mode of actions. So this new fungicide was produced by a company in Japan, yeah, I wonder, you know, which organization would be the one who should take responsibility for this this problem. It's clear that through publications there's more awareness about this problem. Iflufenoquin is now undergoing assessment in Europe. And there's concern in the authorities in Europe of this problem of cross-resistance and the possibility of selection. So you could say, well, maybe we should look at risk assessment and include Aspergillus and other fungal pathogens in the risk assessment. So that might be a strategy. At the same time, we have to acknowledge that if you look at the azoles, the 10-repeat-34 and 10-repeat-46 have never been produced under laboratory conditions. So I think it might also be quite difficult to actually predict the mutations which will emerge. And that would mean that if you find cross-resistance in the lab, you might have to consider not developing this drug either for human medicine or for agricultural use.
Malcolm and Rennes, do you have something to say about the British situation? Because you don't have tulips in Great Britain, isn't it? And Paul,
important from the Netherlands, I think.
Yeah, and you mentioned on one slide, avoid azole, but what about avoiding tulips in Netherlands? Is it a solution?
No, because tulips make us happy.
Hi, Paul. Thanks. Yeah, I think one of the interesting things is that what has happened now is that we are not allowed to put any flower waste, sort of cut flowers like tulips, into the green recycling bins. So they are basically bagged with your plastic and other things and put aside. So they're not taken to the composting facilities. Who follows that is another question, but in principle. Other than that, I don't know. I think it's interesting that this is happening now and getting, happening again and again, whilst we've just been through our CPA patient data and are planning to update the paper we published now over 10, 12 years ago about the trend in CPA patients. That has plateaued. And the way we do stewardship with measuring levels and being very proactive about how we use the drugs in CPA, that is not getting worse. But we see far more in resistance in other isolates, non-CPA patients. So it tells me that the stewardship we do in healthcare does something, works, but what we do outside healthcare, I don't know. Yeah, really worrying.
Yeah, so I agree with that. So I think one of the problems is they want to achieve circular agriculture. So you want like zero waste, and then within this circular use, you could have accumulation of toxic compounds and also resistance. So that's, I think, a concern. And the other concern is that several people, including Bart Vrije, have found resistance not only to azoles but to other fungicides. So if you reduce the azoles, I mean, you won't get rid of all the selection pressure. So I think that's another concern we have to consider.
Okay. So thank you very much, Paul, once again to be with us this afternoon. And now it's my great pleasure to welcome Maiken Arendrup. Everybody knows Maiken Arendrup from Copenhagen and leading UCAST for many years. Yep. So thank you very much for your talk on Candida and resistance. So
that's
you have
true. So
yeah, you can.
It's not this one.
This is a pointer, but to move,
okay, so I have to be able to manage two things. Okay, first I want to say thank you very much for the invitation. It's definitely a pleasure to be here and an honor. So I'm going to talk about Candida resistance to antifungals, and I thought I would do that by first talking about intrinsic resistance. So which are the species that have an intrinsic resistance so that they're not fully susceptible to all the different drug classes. Then I will talk about acquired resistance, and finally I will talk about if we have opportunities to detect this resistance molecularly rather than doing antifungal susceptibility testing, because some people think that is troublesome. So we start out with intrinsic resistance, and Intrinsic resistance is, of course, a consequence of the different species not having the same susceptibility. And therefore, any patient can have such a resistant infection, but of course they will occur more commonly if people have had prophylaxis, because that will select for those that are not fully susceptible. But basically, when we talk about intrinsic susceptibility and resistance, then this can be diagnosed simply by having a correct species identification. I've tried to do a bubble diagram here, where I've put the different species in the bubbles, and I will see if I can point here. I should maybe point here. I don't think I can point. Yeah, I can point. No, I can't point. It doesn't work on the screen, does it? No. Okay, so then I only need one. Okay, but for you that are here, you can see the bubbles here, and this bubble contains the species that are susceptible to candins. This one, the species that are susceptible to fluconazole, and the ones that are susceptible to amphotericin B in the bottom. And in the middle, where these bubbles overlap, you see the species that are susceptible to all three drug classes, and actually that is only these four. So basically, most of the species that we have some sort of intrinsic resistance to one or more of the agents that we have to treat with. You can also see that I've put parapsilosis here, and I've been a little bit ambivalent here about where to put it. And that's because parapsilosis has an intrinsically higher MIC due to an alteration it has in the target gene that is inborn, so to say. So the MICs are higher, but it is a very low virulent organism, and that means that you can basically treat with candins even though the MICs are higher, simply because it's so low virulent. But you do see more breakthroughs, and you do see more persistent infections with parapsilosis. But otherwise, you can see also that I've highlighted some with bigger fonts, and that is, of course, those that are more common. And there's a huge number of isolates that move out of fluconazole. We have also some that are resistant or only susceptible to amphotericin B, and some that are only susceptible to the candins. Now, for many of these rare species, we don't have any clinical breakpoints, because if you want species-specific clinical breakpoints, we need clinical outcome data based on the MICs, and we will probably never have that sort of data for the weird and rare species. So we've done a publication on on a pragmatic approach to susceptibility to classify these more rare yeast, and done that by looking at MICs in our laboratory, verified them across MICs published in the literature, and then we have compared the upper MIC limit of these different species to the more common species that we know, and where we know if they are susceptible or not. And finally, we've looked into the clinical guidelines. Is this drug recommended for this weird species or not? And by that, we have classified them into 3 groups: treat if wild type, consider use if the disease is not severe, if you can use an elevated dose, if you use it for all consolidation, or if you have no better option. And then we have a group down here that says consider alternative therapy. And I hope you will find this useful, because many people often ask us what to do with the MIC of an organism where we don't have any breakpoints. But there are this number of isolates that are not fully susceptible, and how has that developed over the time period? How big a problem is that? This is the Danish surveillance program, which has been ongoing since 2004. Where we've had all blood culture isolates from every patient in Denmark. It's easier to do when you're a small country. But I think you can appreciate the bars are the number of infections, so there's maybe a little bit of an increase over time, over this time period from 2004 to 22, but you can definitely appreciate the Candida glabrata has gone up. It has really increased over that time period, and that is on the expense of Candida albicans, which has gone down. And actually, when I was a medical student, albicans was 85% of the isolates, and glabrata was even lower than we have here. But today in our country, they're almost the same, and around 35%. We even had a single Candida auris case in 21, but it has not been a big problem with us. But if you move to a study covering Europe, then that was initiated by Martin, who's here on the first row, where we collected isolates from and also clinical data from patients in Europe. And each center had to contribute with at least 10 isolates. And here I have included the countries where there were at least 2 centers, because I think doing a species distribution only on 10 isolates, that's definitely too small. And then I've sorted the different countries according to the albicans prevalence. And what you can appreciate here is that there's quite a difference over the countries. So Austria, for instance, is a country where we have a traditional, so to say, species distribution with 70% being albicans, whereas we in countries like France, Turkey, Greece, Spain have much lower albicans rates at expense of other species. You can also see that in the north, in our country, which is not on this diagram, but also in Sweden and in Belgium, and I know that's also the case in Norway, we have our second most common species is glabrata, whereas in the more southern countries like Greece, like Spain, for instance, Turkey, parapsilosis is the second most common organism. So there will be differences across our different countries on how prevalent these species are, and therefore also the intrinsic susceptibility pattern, the local epidemiology that we should be prepared for when we treat our patients. All the colors that are dotted, like the glabrata here, and the dotted up here, are species where we know there is some intrinsic resistance. So even though it may be difficult for you to say, but then there is quite a number of dotted species on top of the glabrata. So I think that intrinsic resistance is fair to say that is also an issue. You can also see that in this study there are very few auris. There's a few auris here, so in total very few. And that's probably because this study was conducted in departments that didn't have the big outbreaks that accounted for the majority of the isolates in Europe. But I want to talk also about auris, because auris is an organism that is phylogenetically related to glabrata. So higher fluconazole MICs from the beginning, but over time, the vast majority of the auris that are circulating globally are completely resistant to fluconazole. So we have two compound groups left, the candins and amphotericin B. And if you look into the literature, you will see huge variations in the resistance rates reported for amphotericin B. Some say 30%, some say 0. And this is compiled from a study here where the amphotericin B MICs, the ranges are indicated with the blue lines, and different studies from different parts of the world. And the red line here is the resistance breakpoint for amphotericin B at 2. So anything that is on this line or to the right of this is resistant. So there's a huge variation. There are some studies that report everything susceptible, as I said, and some everything resistant. And then one of these studies tested the same isolate with 3 different methods. So here are the test results using the CLSI, the American reference method, the broth microdilution method. This is using Vitek, and this is using e-test. And you see how these MIC distributions are completely different with the different methodologies. So the resistance rates for these identical isolates goes from 1% to 100%, depending on the different test method you use. That's really not acceptable. So we tried also to look at different methods and included a strain collection that Sean Lockhart has at the CDC. So it included all the different 5 clades that circulate on our wonderful planet. Sean had tested it by e-test when he received the isolates. So that was one isolate on one day, another isolate on the other day, so many different batches of plates, of technicians, of everything, and he had a huge variation in the MICs going from 0.03 to 16. We repeated the same isolates using e-test, and ours were more narrow, but that's not because we are better, it's because we did everything with the same technicians in a short time period. Then we have used actually what they say you shall do when you do e-test, and that is to use a higher inoculum than most people use. And that moved the distributions a bit to the right, particularly if you read them after 48 hours. Then Nathan Wiederhold tested them with a different MIC e-test strip. And finally, we did UCAST, which collected the isolates here, and CLSI broth microdilution repeated twice. So if you look at the resistance rates here, it goes from 0% to 63%. And that's the same isolate. I mean, that's not meaningful. So basically, there are two challenges here. There is a huge test-to-test variation, and there is the other issue, and that is that if you look at these isolates, then we have for UCAS, we have the majority of the isolates here, and that's also true for CLSI. And the breakpoint is here. So that's just, it's simply at the upper MIC we have. So a minor variation will move things around here and impact the number of isolates that are classified as resistant. So the breakpoint is simply too close to the wild-type distribution. Now there was a different study that, and now nothing happens. I'll try once more. I can't move it forward. Maybe this one is, maybe there's no, now it moved. Yeah, but I think that was a technician. So I have to say next if it doesn't work. Now, this is also found by others. This is isolates that is from the Walters group, tested by e-test here and tested by TREC. So you can see, again, a huge variation between the two methodologies. You can also see a nice Gaussian distribution here for the e-test. It's isolates from a single patient. But there are 4 isolates here that then would be classified as resistant if you use the breakpoint. Now, these isolates were whole genome sequenced, and none of them harbored an alteration or mutation that is known to confer amphotericin B resistance. When we collect isolates and do MIC testing, there is an inherent variation in the test, and that is independent on which test it is. We showed that quite some years ago for glabrata, when we had clinical isolates here, and in parallel with testing these isolates that ranged from 2, as you can see, and up to greater than 16. We took the first isolate with an MIC of 2 and tested that repeatedly 51 times every time we tested also a glabrata. And this is the MICs of that single isolate. So you can appreciate that it mirrors exactly the distribution above. So that means that what we have within here is test variation. So it is, it's a single isolate. It has the same MIC, but this inherent variation in a phenotypic test. That's not typical just for Candida testing. It's exactly the same for bacterial testing. Here you have a Staphylococcus aureus that is tested by disc diffusion. So this is zone diameters. This is a QC strain that is tested a huge number of times, and this is clinical isolates that are tested, individual isolates. So these mirror one another, so that is the same. And this, of course, means that if you put a breakpoint here, you will have a random classification of your isolates that has an identical susceptibility. So that's definitely a no-go. So we basically have to decide if we believe that Candida auris is susceptible or not to amphotericin B. And if we compare it to Candida glabrata, because it's phylogenetically related to glabrata, you can see there is a little bit of a difference. Auris is a little bit, has MICs that is a little bit higher. Here you have the mode for glabrata here, but this is it's almost the same. So you could say the mode is somewhere in between here, the most common MIC. So there's 1.5 step difference. So we have to decide if we think this is susceptible, and if it is, we have to put a clinical breakpoint that doesn't bisect this distribution and will include and allow also a little bit of variation going up to 2. So probably 2 would be okay. Or we have to decide we don't think we can treat with amphotericin B, and then we have to deem it resistant. Because currently, if you use any kind of commercial test or whatever, you can have a completely false result. And I think that's important because Candida auris is an organism that can also quickly get resistant to candins. Our first patient in Denmark was a patient that was just colonized, so this patient never ever had any antifungals when she stayed in the hospital for a month. And then she was discharged, and there was sampling and everything, and there was cleaning, and the room was left for 24 hours before the second patient was admitted to that same room. And that patient had no travel history, no prior use of antifungals, and stayed there for 5 hours before he left for a second hospital. And at the second hospital, he had a Candida infection, so he had an ill function for 14 days, so 2 weeks of Candidas, and then he returned. And at the day of when he returned to the first hospital, without any antifungals, he had fever, taken a blood culture, and then he had this auris isolate. And you can see here by genotyping that it was exactly the same genotype. So he did get it from the first patient, even despite we had cleaned everything the best we could. And you can also see the only difference now is that this isolate is Candida resistant. So it took exactly a max of 14 days. So if we deem amphotericin B out, then we have to be sure that's correct, because otherwise we have very little to treat it with. This patient actually got amphotericin B, and he recovered, but he was still colonized after 1.5 months, but not in his bloodstream. Okay. So with that, we'll move to acquired resistance. And acquired resistance is, of course, when we have a species that normally is susceptible but suddenly become resistant. And here, a species identification cannot tell us it's resistant. We have to do some sort of susceptibility testing. How frequent is that? Where are the main problems with acquired resistance in Candida? And I will say that basically, it is much more difficult to get resistance in Candida than it is in bacteria. And that's probably because we don't have plasmids that are exchanged, so every resistance has to arise in that isolate. But we do have some areas where we have an issue, and that is, one of them is in Glebata, where we have 10 and 14% resistance on top of the lower susceptibility that Glebata has in the ECMM and the Danish surveillance study. So that's quite uniform, and that we see this level of resistance in Candida globata. What is not uniform is that in the ECMM study, there was a resistance rate of fluconazole resistance in parapsilosis of 21%, and we have a rate of 2%, so that's a huge difference. And this is due to particularly one genotype that has a resistance in the ERG11 target gene, a Y132F alteration that confers resistance. And that has been detected in our study in Spain, Italy, Greece, and Turkey, and since that also in France and Israel. And you can see on the map here, and of course also here, that has also been detected in North and South America, in South Africa, and in Asia. And I think that is problematic because these strains have caused outbreaks, and parapsilosis is a strain that very easily can be conferred from one patient to the other. So this is something that you have to take into account both for traveling and for these centers where this is endemic. When we look at acquired echinocandin resistance, the rates are much lower. Here, and they're quite similar here in Glebata across the ECMM study and the Danish surveillance study, around up to 2.5%. Now, this rate is not very high, but it's probably, or perhaps at least, you can say it's the tip of the iceberg, because we do know, and there are several studies that have shown that there are more resistance outside the bloodstream than in the bloodstream. If you look at isolates, for instance, in the mucosal surface in the oral cavity after end therapy, and you look at the patient that had at least 7 days of Candida therapy, then the resistance rate is really high in Glebata in the oral cavity compared to the bloodstream. This is a Danish study, so it's the same patient, and the rate here in the bloodstream and the rate in mouth swabs after end of therapy. And Rennes in Spain has done a nice study where they looked at samples from sterile foci in the abdomen and what the resistance were there compared to what the resistance rate is in the bloodstream. So again, higher in the deeper and more difficult to reach foci than in the bloodstream. And what could be the reason for that? Well, I think we should consider that candins are huge molecules. They are bound to proteins, so they don't really penetrate extremely well into these kind of areas, deep foci or biofilm or oral cavity. And that means that we will have subtherapeutical levels of candins there, which is a known risk factor for selecting resistance. And these isolates can, of course, be a source for subsequent infection that is then more difficult to treat. And I think the final issue that is emerging is that we now also see some acquired candida resistance in parapsilosis. That is the organism that has high MICs already from the beginning, but is very low virulent. But now we have seen isolates that are completely resistant due to target gene alterations, both in Greece, in Turkey, and in China. And in the last two of these, it occurs in isolates that are fluconazole resistant. So now there's only amphotericin B left to treat those. And I'm sure we can agree that we don't want these kind of isolates circulating in our ICUs among patients there, right? Okay, so what about the mechanisms and how easy would it be to detect isolates molecularly? And what about timing? How quick is resistance emerging? And I've taken this case from Denmark as an example, where we had a patient that had an esophageal cancer, so therefore he had radiation therapy, and therefore he had esophageal candidiasis. And he was treated first with azoles. You can see they are marked in blue here. And he was treated with fluconazole for a year. And then he had an organism that was fluconazole resistant, and that is indicated by the shaded area here. Then he was changed to voriconazole and had Voria and Posa together for almost a 2-year period. And then he had an isolate that was completely azole resistant. Then he had caspofungin first in 1 and 2 weeks, and then he had alternating caspo and nilafungin for 5 weeks, and then he had a candin isolate that were both azole and candin resistant. And if you compare the time periods here, it takes almost like forever to induce a resistance to azoles, where it's actually quite short-term courses that we need to select for resistance for the candins. And finally, he received polyene therapy, both amphotericin B and nystatin, and guess what? He ended up with an amphotericin B resistant organism. And if you look at the MICs and at the underlying molecular alterations in these isolates, then you can appreciate the quality control strain up here in albicans and the first isolates that were completely comparable to the control strain. And then we had the first isolate that were fluconazole resistant, and it had 4 alterations in the ERG11 target gene. These 2 at least have been reported to cause fluconazole resistance. And this isolate also had some elevation of the CDR2, which is an efflux pump that pumps fluconazole out of the cell. Then he had the isolate that were pan-azole resistant, and here was an additional alteration in the target gene that has also been described before. And we had a marked elevation of the CDR2. Then there is the isolate that is caspofungin resistant. It has a well-known hotspot alteration. And finally, we have the isolate that is resistant to everything, and that has a homogeneous stop codon in ERG2 that is associated with amphotericin B resistance. But this illustrates that it will be difficult to do a molecular test for azoles, at least in a clinical setting, because it's multiple actions or mechanisms that play in concert. So that's also why we, in the UCAST paper on recommendations on molecular methods, say that there is no assay and there's no commercial assay, no in-house assay to detect azole resistance. And that's due to these multiple mechanisms playing in concert. But of course, if you are in one of the centers where you have this outbreak strain, then you could do a PCR test simply for that, and that could be useful in those centers. But for the candins, all the resistance we know so far are due to hotspot alterations in the target gene. And this is listed on this slide, where we have the different species out to the left and the normal upper limit of the MICs for wild-type isolates. And then we have the 2 hotspots in the target gene FKS1, and also the 2 hotspots in the second FKS gene that is also in play when we talk about resistance in Candida glabrata. And there are these 2 hotspots. This is the amino acid abbreviations, 1-letter codes. And where this letter is red is typically where an alteration would give a high-level MIC elevation. Where it's yellow, it will give a more moderate MIC elevation. The green ones do not impact the MIC at all. And this is the alteration that we see in the psilosis group that causes the higher MRCs that you see in this group. So you can sequence, of course, and you can see if you find any of some of these mutations, you have an idea on if they confer high or low level MRC elevation, even though there are also some differences depending on which amino acid is replacing the original one and in which species it occurs. But life tends to become more and more complicated all the time, and basically this is also true here. So there are 2 new publications on alterations outside the hotspots that also confer resistance. So with that, I think the take-home messages are that resistance in Candida is, of course, an issue. And there's an issue with intrinsic resistance in Candida glabrata and in Candida auris. And then, of course, there are all the rare species that are rare, but that we meet sometimes, and many of which have a reduced susceptibility to one or several drug classes. Regarding acquired resistance, the main issue is for fluconazole is resistance in glabrata and in parapsilosis. And for candins, Candida glabrata and auris again is the problem, but it's also concerning that we now see it emerging in parapsilosis. For Candida, it's dominated by target gene alterations, where codon species and substitution impact the MRC elevation. But there are also new alterations outside. And therefore, there are, of course, alternatives to phenotypic testing, particularly for the candin resistance, but I think it's important also always to remember that these tests can detect resistance, but they cannot confirm susceptibility, because you could have more than one isolate, you could have new mechanisms that you were not aware of. So with that, I want to thank you for your attention.
So thank you so much, Maiken, for this fantastic talk. So do you have some question? I have one regarding variation test. What is the duration of incubation do you recommend with e-test? Because when you are reading e-test, particularly with Candida glabrata, at 24 hours or 48, it is sometimes far different with 2, 3, 4 dilutions of difference. So what is recommended for you?
Well, the tests say that it should be 48. You should always have the 48, but I think that will cause false resistance. Yeah. And that was definitely also true for Candida auris. Yeah, definitely. And
I completely agree with that.
Yeah, I would say we did because most of the isolates from the hospital go to SSI, where we do UCAST, but we use e-test at the hospital also. So we had a training course where we did a blinded collection of wild-type strains and mutant strains, and tested that, and the results after 24 hours were basically those that were clearly best. Mm-hmm. But it can be difficult if you accreditate it. Your laboratory and
that's difficult, but that's why it's important that national medical societies, national and international,
give some advice on this.
Exactly.
Exactly. Yes.
Yeah. Thank you. Fanny, do you want to say something regarding Candida albicans? Because in the ECMM study, the level of Candida albicans was lower than the national survey. The national survey is around 45%. I think.
Yeah, so in the national survey we have, so the number of Candida albicans was 40%, so which is quite higher than, because that was selected hospitals and probably there's a bias.
I think that is clearly the caveat with a study like this, that it doesn't capture necessarily the nationwide rates. That's true.
Yeah, so absolutely. I was just checking, and it's 44%.
It's also funny to see actually that there is a huge difference among the centres, and in some countries they were almost completely the same. So the 6 centres in Germany, or how many it was, they had exactly the same rates. Whereas in Great Britain, they were all over the place. I mean, there's a difference in mentality, I think. Yeah. Whether you stick to guidelines, you're supposed to do that in Germany more than we do that up north.
Okay, thank you. The last question, because after it will be time for the next talk.
Thank you for the talk. What about the acquisition of resistance of Candida tropicalis, especially in Asian areas?
I think that's a very good question. The question was tropicalis and resistance. And one of the issues with tropicalis is that it has this trailing phenotype often. Trailing means that it has some residual growth in all the wells, also in the higher concentration wells, and that can interfere with the endpoint reading. So I think Tropicalis can be resistant, but there are probably also some impact of this trailing phenomenon in different studies, which may give you the message that there's more resistance than there truly is. We put isolates, trailing and non-trailing, in a mouse model to understand whether, what that means. Because sometimes when you do UCAS testing, you have a spectrophotometer curve, and some of these isolates, they are almost up to 50% growth, or they are a bit higher. So we took isolates that were trailing a little bit, that were trailing around 25%, just below 50%, above 50%, and completely resistant at 100. And then we put them first in larvae, and we had exactly the survival curve that matched this stepwise increase in trailing. But when we put them in mice, we couldn't reproduce any difference in susceptibility unless they were above 50%, even when we put them in immunosuppressed models. So based on that, at least in our laboratory, we still report isolates as susceptible if they trail only below 50%. But if you do the CLSI methodology, I don't know if you have seen that, then you look into your wells and you look at the growth, and there's just growth all over. I mean, it's impossible to do that. It's easier in the UCAS because you simply have a growth curve, and that's objective. It's compared to the control of growth of the same isolate in the plate.
Okay.
Yeah.
So, thank you very much.
Thank you very much.
And now we can move to the next presentation with Patrice Le Pape, who is professor in Nantes University, and he will talk on fungicides in the environment. Yeah, okay.
Okay. Well, thanks, Jean-Pierre, for the invitation and for the introduction. Dear colleague, good afternoon, or better, good evening. Yes. And so, you know that for today, for all the people working on antimicrobial resistance, it seems obvious that and that we need to resolve the problem and One Health approach. This is a fact for our antifungal diseases, since given the link between the use of fungicide and the driven environmental resistance, which was suspected by the group of Paul Verweij as early as 2009. This side effect that is mentioned in this paper is of concern when we see the predicted evolution of the fungicide market, which reach about 28 billion by 2027. And it's important to note that the highest growth rates could be provided by an increasing demand in developing countries and the growing area under high-value cash crops, such as fruit and vegetable. And you know that this country and this sector, fruit and vegetable, have been little investigated for other resistance and could be the next challenge for our research. Among the numerous family of fungicides, strobilurubin, carboxamide, benzimidazole, morpholin, the azole family, called DMI for dimethylase inhibitors, are the most important in terms of launched molecule in the market, but also the most used molecule in the agriculture due to the broad spectrum and the persistence in the treated fields. Oui? Okay. The responsibility of this azole fungicide in the resistance has been suspected on 3 major elements. First, the structural homology with medical antifungal drugs. You can see that the agriculture azole have azole ring, triazole or imidazole, that can bind to the M of the demethylase, and an heterocycle substituted by 1 or 2 halogen that can bind to the tunnel or the hydrophobic pocket of the enzyme. The second is a publication in Penicillium digitatum, a citrus pathogen treated by trifimazole or imazilil, it's also imidazole, of the same tandem repeat mechanism that described in Aspergillus CYP51 promoter. It's a 126 tandem repeat instead of 34 or 46 for Aspergillus. The third factor is a confirmation by our team of environment as a source of azole-resistant infection in a patient which is a farmer who died by cerebral aspergillosis. And the environment and strain of Aspergillus were indistinguishable from the patient isolate. We can note that in the garden and the home of the patient is front of the cereal crops, which were treated by tebuconazole. So it was legitimate to think as Aspergillus are ubiquitous fungi together with targeted plant pathogen in the soil, that exposure to the azole fungicide causes this type of genomic alteration. Well, what are the indications of the DMI fungicide? The indication range from the protection of crop production and quality to wood preservation against degrading fungi. Concerning the cereal production, you can see that the growers lost up to 30% of their cereal crops, and in the state of unit state, and up to 50%. And the fungal disease are fusariosis, septeriosis also, with Mycosphaera graminicola as a major pathogen of the wheat crops. In Europe, fungal disease are most problematic in France, Germany, and UK, explaining the large reported azole use in these three countries. And for France, you can see that it's a humid climate of northern France that reveals the sort of azole fungicide. Another use of DMI are the preservation of flower production. With the talk of Paul, the Netherlands and Colombia are the 2 largest exporters of flower in the world, and Colombia is ranked 4th in the world for the use of pesticide. In this study that we have conducted in Colombia, where flower fields, greenhouse, ornamental flower beds were sampled, azole-resistant isolates were identified in about 10% of the soil sample. And this resistant isolate arbored not only diverse mechanism of resistance, but more interesting, a great diversity of genotype, indicating an active reproduction in the soil. 4 triazole fungicides were detected in the same soil sample, which allowed us to cultivate azole-resistant strain, giving a new in situ argument for association between triazole fungicide and Aspergillus resistance. One of the epidemiological facts that it's very interesting is the possible geographic transfer of azole-resistant Aspergillus fumigatus with flower bulbs. With these two publications, a transfer from Netherlands to Ireland and a transfer of azole-resistant Aspergillus to Japan. The DMI are also used for the vegetable production, protection of vegetable production with this pathogen, this Phytophthora infestans, this fungus-like microorganism, is responsible of the late blight of potatoes and other vegetables. And if you remember, is responsible of severe famine in Europe, leading to immigration of a part of the population of Ireland to the USA. In this other studies that we conducted in Cundinamarca, some vegetable crops were sampled. These vegetable crops were treated with tebuconazole and difluconazole, and you can see that this sample exhibit a large quantity of azole-resistant Aspergillus strain. Okay. In the crop protection, there is also the fruit in the orchard. The high water contents of fruits make them particularly vulnerable to fungus attack by Alternaria, Patrice, Penicillium, and we can see that for Monilia fructicola, that it's an agent of brown rot, they need for the control more than a dozen of applications that lead to a concentration of the azole in the great concentration of azole in the soil. Okay. Well, we stay in the crop consideration, but not for protection, but a use of azole as plant growth regulator. They are inhibitors of gibberellin hormone, and they increase the number and weight of fruits per tree. And so this is an example of pacrobutazole that is used in culture of a mango that we have investigated with our Brazilian colleagues. Well, we move to the crops fields to another indication of the DMI is the preservation of wood against the sap-stained fungi, and we can see that in the dipping technique to treat the wood, that this technique often leaves a quantity of azole molecule in the ground after the drying, hot drying or air drying process. In this context, a number of lancet products containing propiconazole or tebuconazole is high, and this indication represents about 20% of the total of the same molecule sold in France. The environment is also a problem in medicine veterinary, and you can see that the enilconazole, that is the other name for imazilil that is used in agriculture. Enilconazole is sprayed to treat litters or beddings in poultry farm to prevent outbreak of avian aspergillosis. And this study, it's a review by Gomez. You can see that in this publication, resided in poultry farm in Portugal, Aspergillus fumigatus is the most frequent species identified from Aspergillus genus. It's interesting to note that the bedding is made of straw, wood saving that contain also azole molecule. And the other point is that these aged beddings or aged litters are also used as manure for the crop field, and so it's a toxic cycle of this compound. Well, you can see that there is a lot of indication for the DMI fungicide, and azole fungicides are widely present in our environment. And the persistence that it's a characteristic of this moderate lipophilic compound increase this presence. You can see that the tebuconazole, for example, has a half-life of up of 600 days. That is very, very high. And that the azole was retained in the top 20 centimeter layer of the soil. That is the localization for the multiplication for the fungi. And so there is a contact, intimate contact between the fungicide and the Aspergillus. The presence in crop soil is well illustrated in this publication. You can see that, as expected, the tebuconazole is detected in the crops field where the tebuconazole is used, but also in the organic farming. And there is another data that is interesting, is that the epoxyconazole is also detected in this crop field, despite the molecule was not sprayed over the 2 years before sampling. So this indicates the high persistence of the fungicide. Azole fungicides are also present in flower soil, as we have demonstrated and commented by Paul. In the sewage sludge, which has been sprayed on agricultural soil, present in suspended matter from river, present in subterranean water. You can see in France with the 200 measuring stations that the tebuconazole is very present. And in our region, present also in the superficial water. And you can see the spatial increase of the presence of tebuconazole in the last year. Well, how does environmental resistance arise in Aspergillus fumigatus? This process of selective pressure depends on multiple factors. The first is the exposure of Aspergillus to azole concentration, which in turn depend on the type of azole applied. Not all the azole have a great affinity to the demethylase of Aspergillus. There is five azole with high affinity. The lipophilicity, which influence the persistence of the molecule, and also the characteristic of treated soil. In this publication of Bromilov, we can see the difference of the half-life of the triadiminol of the epoxyconazole, which differ in the clay substrate in comparison to the sandy counterpart. The fungicide professional practice influence also this resistance in terms of frequency of application, presence of mixture of compound. In this publication of Bastos, you can see that 2 or 3 spray with an interval of a week is sufficient to lead to a high concentration of propiconazole. We need also conditions favorable for reproduction and abundance of Aspergillus, just as organic matter, temperature, humidity. There are conditions that we can encounter in wastes from flower bulbs, green material, wood chipping, as commented by Paul previously. And this, with the compost, are environmental hotspots for the resistance. Well, the prevention of azole-resistant Aspergillus emergence or study, next studies, need some information. First, the resistance risk must be assessed separately for each hotspot in every country, but also, I think, in every region of a country, because we have a great difference of crop culture or indication for the DMI fungicide. We have to see also the indirect effect of regulation of authorization of use of this compound. For example, the ban of some azole fungicide identified as endocrine disruptor, and this ban is ongoing in France for epoxyconazole and for two other azole fungicides. Change in agricultural practice, we can mention the discontinuing DME application which is presented in the Suzuki publication that demonstrated that the discontinuing of tebuconazole lead to a reduction of tebuconazole-resistant Monilia fructicola after 2 years of discontinuing. Well, we have to make caution also to the introduction in the agricultural field of higher active triazole fungicide. That is the case for the methylphentrifluconazole. This molecule was launched 3 years ago, and it's very active against the phytopathogens that are resistant to the conventional triazole fungicide. In our docking model, we demonstrated that fit well with the 14 demethylase of Aspergillus, and this molecule could lead to a transient diminution of the resistant Aspergillus in the field, but only a transient, because we can expect that there is a new mutation in Aspergillus, in the Aspergillus strain. And the final is the need of a great program for identification of new fungal targets and new medical antifungal drug to counteract this Aspergillus resistance. Is a case of Olorofim, that is the next talk, and the derivative of this molecule that are ongoing in the market by Japan industry for treatment of the crop. And so it's really a great program for the management of this resistance. Thank you very much for your attention.
Thank you, Patrice, for sharing with us your experience in Colombia, in South America. So do you have questions? I have one regarding, because we now know the problem of the resistance of Aspergillus fumigatus particularly, but what is the impact on other molds, mainly particularly other Aspergillus or Fusarium?
Yes. With Aspergillus flavus, there is a great experience in South America, because it's the most frequent Aspergillus, species Aspergillus in this continent, and there is also an increase in the MSI of flavus against the azole molecule, and so the problem is the same. Perhaps the hotspots are different, because the presence, the hotspot for flavus are different from fumigatus. But it's the same problem. And perhaps for Oris also, because if Oris is resistant to fluconazole, perhaps there is also an implication of an involvement of this fungicide in this yeast.
Thank you for your talk. It was brilliant. We have also, over the past 3 years, done a lot of hotspot sampling in Denmark, and we have revisited several of the sites that we have, sampled from the initially, and seen quite some variation. So I think that to me, it's quite difficult to actually say how can we get a uniform understanding on the different rates, the different places, because temperature, humidity, how deep you go in soil, when is last spring, but particularly the climate things, they impact the number of fungi, the Aspergillus that you actually culture. And we had the first summer season, we didn't have very much Aspergillus. The next summer season, we had a lot more in our samples. And of course, if you have a rate, as we do in Denmark, around 3.5%, in our samples, which is also what we find in the patients, by the way, so it's probably realistic. But but then you need to have at least 170 isolates in order to have a 95% confidence in detecting one mutant. So, I mean, you need to test huge number of isolates, and just some variation in humidity or whatever in these areas where you sample, which are definitely not homogeneous. I think it's really difficult.
Yes, yes.
That was the first question. And the second one is, what do you think about the free fraction versus the total of the DMIs? Because these DMIs, they bind very strongly and extensively to the soil and are very difficult to release from the soil again. So I think also that it's difficult to understand what type of procedures are and when is the selection occurring. And I'm curious to know if you have ideas on this.
Yes, because these compounds are high lipophilic. Yes, and so this compound bind to the matters in the soil. Yes, and the real question is how is the concentration that you need, free concentration that you need to make the pressure to the fungal Aspergillus, and perhaps it's not the high concentration, but perhaps it's only the low concentration and the variation of this concentration in the time that can make the pressure for gain for resistance. But there is really no study. Yes, 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 and yes, yes.
For the agricultural fungi, they say it's a high concentration.
Yes.
Tend to believe it's more low concentration.
Yes, yes.
But also the concentration you measure in your soil samples, that is at that day when you sample.
Yes,
it's not necessarily the same the next day or the next day, because there's springs, a few times. So how do we actually understand and get an understanding on when and why? We've not been able to reproduce selection in soil. We thought that would be pretty easy to spray, to culture, to have a mix of wild type mutants. We couldn't make them grow even.
Yes. So there is some publication about the production, in compost and to in vitro production of resistance in vitro in a publication from China. I don't remember the author, but it's very difficult to make that in the lab. We have not realized this experience with good result in the lab for paclobutazole, for example. Yes, and the other problem is that when we make the extraction of fungicide in the soil, we are measuring the bind and the free molecule, and so it's very difficult to discuss. And after it's only a photo at the moment, yes, and no, it's not sequential because there is really few publication with a sequential demonstration of the presence of fungicide. Yes, it's okay.
Thank you. Another question?
Yeah, thanks for your talk. Something I was wondering about, because I saw it in your recommendation, and I think we always tend to say that we should use less azoles in the environment and we should limit our use. But given the fact that they have a really long half-life, and for example, in wood preservation, it lasts for a really long time, up to 30 years or something, and the fitness difference between the resistant and susceptible Aspergillus is not that much difference in a non-azole environment. What percentage of the resistant fraction do you think will decrease if we stop using azoles in certain practices? Do you think it will have an effect or a significant effect? Or?
Well, we have no data for Aspergillus, but if we compare with phytopathogen, the resistant phytopathogen, we need about 3 years to see a decrease, a real decrease of the resistance.
Yeah, but that's probably because there is a fitness difference between isolates when there is no fungicide present, right?
Yes.
Yeah, but it's not the case.
But for Aspergillus, this tandem repeat alteration give not a fitness.
Yeah.
Yes.
Yeah. So does removing the azole remove the resistant isolates from the environment?
No.
Yeah.
No.
So yeah.
Yes.
Okay, thank you. Last short question.
A short question and a comment. We haven't really talked about eutrophication, the washing out of soil into rivers and ponds. I mean, fertilizers are known to end up in water distribution systems, basically ponds, rivers, then obviously purification of that water. Do we know much about the levels of these azoles in ponds and rivers and in water distribution systems that end up in our hospitals, end up in our homes?
There is a really low concentration of azole in the superficial river and in the water. Yes, but if you accumulate all the detected molecule with the same mechanism of action or mechanism for resistance emergence, the level could be high. Yes, but in the water that you use, there is by this kind of purification, we have not detected the azole. Other pesticide, but not the azole.
Okay, so very interesting topic, many questions. Thank you very much, Patrice. And now we are online with UK, with Emma. Emma, can you hear me?
Hello, good evening. Yes, I can hear you. Can you hear me?
Yes, perfect. Hello, Emma. Nice to have you. We miss you in Rennes.
I'm sorry I'm not there in person, and John Rex also shares his apologies, but he is on a flight back to the United States and so not able to join.
Yes, okay. So with Martin, we're going to share this discussion. We had a very nice session on antifungal resistance, Aspergillus resistance, Candida, the use of fungicide. And we wanted to have the example of the development of a new drug. And you are going to tell us how does it work to develop a new drug. It's not so easy, but so please start, and then we will have some question after.
Of course. Well, thank you very much for the invitation to speak to you. So it's a pleasure for me to talk to you about the case example of Olorofim, which some of you may have heard of. And just my disclosures, I'm an employee of F2G, and F2G is the parent company that's been developing Olorofim, and we're now doing the development in partnership with Shionogi. So I'm going to cover three topics fairly briefly: general challenges in developing and for invasive fungal infections, Olorofim as an example, and some of the lessons learned. So one of the major challenges in R&D for invasive fungal infections is, of course, that fungi are eukaryotes. So finding a druggable target that doesn't actually lead to significant human toxicity is a major challenge. There are very few targets that are unique to fungi. Novel mechanism agents are therefore rare, and of the novel antifungal agents in development, most are actually adaptations of drug classes already available. For example, new formulations of polyenes, new formulations of azoles.
Sorry, Emma, can you switch the format to the full screen?
Oh, I'm sorry, apologies. Let me just check.
Display settings.
Sorry, I'm looking at the wrong screen, I think. Nope, it's the wrong one. Sorry, I cannot currently see the right taskbar.
So perhaps stop sharing the screen and then your display settings and
Apologies, let me do that last. Go back. Is that giving you the right view?
Yes.
Okay, yes.
Perfect. Thank you.
Okay.
Now it's coming.
Apologies for that. It's always finding the right button. Different systems are always give you different options. So coming back to the slide, when you're looking at non-clinical efficacy models for antifungal development, those models are limited and they are limiting. So mortality alone is not a great tool. You need a continuous readout of data to support PK-PD relationships. So if you're killing your animal models, that's not a great tool to use. Of the well-known and established animal models, very typically Candida, Cryptococcus, and Aspergillus can be done, but actually well-established models for other fungal infections are not really present or not as readily present. And using rodents, particularly mice, for these studies also presents challenges, as rodents are not small humans and they don't metabolize drugs in the same way. Preclinical trials are also very long running and they are expensive. So in order to be able to move into human studies, you must be able to do to dose for at least 6 weeks, if not longer. So the animal model, 6 months, if not longer. So these studies take a long time. So to permit dosing in man, in vivo animal model studies must span relevant treatment periods in at least 2 animal species. So we have to do up to 90 days of exposure to allow for equivalent dosing in humans. And you cannot start human studies until you have done a 90-day animal model study. To allow then for chronic dosing in man, you need to do dosing studies of 180 to 270 days, and those are typically again in 2 models, very often rats and monkeys. And this has significant implications for the time required to run these safety studies and for the cost and duration of human studies. So you can do acute studies if you have 90 days of animal model data, but knowing many of these fungal infections take a long time to treat, you actually need the chronic toxicity data before you can go into chronic treatment. And you need a lot of drug substance to do these studies. We're talking kilograms of drug substance. What about clinical trials? Well, these are also prolonged and also expensive. So invasive fungal infections are rare. They qualify under orphan disease categorization. So there are very few fungal species that actually can be studied in a fully powered non-inferiority randomized controlled trial versus a modern comparator. So randomized controlled trials are possible in Candida, in Cryptococcus, and Aspergillosis. They are much more challenging to do in other mold infections, so mucormycosis, Lamentaspora prolificans. These would typically require an underpowered or even unpowered study design. And these studies run very slowly. They take years to do. Patients are infrequent, they're difficult to diagnose, and not every patient meets the very restrictive entry criteria for a clinical trial. And also, you're looking at doing studies typically in patients who are already on other therapies. If you're talking about immunosuppressed patients, then you need to understand potential drug-drug interactions. You need to be able to manage those. And you always have the confounding fact that mortality is not always down to the invasive fungal infection. So trying to tease out cause of mortality from the IFI or the underlying disease is a challenge. So I have to thank Johan Martins for letting me use this graph. This is just a schematic of the major studies that have been done in invasive aspergillosis over the last 25 years. So starting from the left, the voriconazole versus conventional amphotericin B study, and moving across to the right to the most recent voriconazole versus posaconazole study. And you can see that over time, the duration of these studies has increased, with the most recent study taking 6 years to report and on average 4.5 years. And now there is a requirement for several hundred patients. The power of these studies has changed. So now there is a 10% non-inferiority margin, and that increases the number of patients. So the latest study, there were 653 patients screened for this study. That's a big number. These studies take a long time to complete, and as underlying therapies change, as treatments for underlying diseases change, actually the study populations may no longer be typical of the patients that we see in everyday treatment settings. So very often these studies will have excluded azole resistance, they will have excluded breakthrough infections with azole prophylaxis, they will exclude the patients where drug-drug interactions and toxicity are likely. So doing a study that actually covers all of the patients at risk of, let's take invasive aspergillosis as an example, is a big challenge. So a little bit about Olorofim. So F2G was actually founded in the late 1990s. It was originally founded to look at fungal genomics as targets. And the first erotomide was actually identified in 2005. The erotomides is a new class of antifungals. They have a novel mechanism of action. The biology required substantial exploration of candidate molecules. And it took from 2005 to 2014 to find a molecule that actually had a reasonably broad spectrum of action and that could be dosed in man safely, or in a as safe as possible a way. So Olorofim entered phase 1 in 2014, and those phase 1 studies ran through to 2018. And one of the reasons that took so much time is that while the pharmacokinetics were very predictable, there was a hepatic injury signal identified. And this was shown to be dose-related. And so a lot of those phase 1 studies were actually repeated, trying to find an optimal dose that would balance potential efficacy with acceptable safety so that the risk-benefit was in an acceptable bracket. The Olorofim phase 2 study, which we've nicknamed Study 32, was a single-arm study in the setting of proven mold infection or probable pulmonary invasive aspergillosis, and in patients with limited or no treatment options. That study completed enrollment earlier this year, but it took us 4.5 years to enroll that study. The benefit risk was appropriate in a salvage population. We've shown that the drug is efficacious across a range of molds with a manageable liver signal and with pharmacokinetics and drug-drug interactions as expected from the preclinical data. And that phase 2 study has completed and the regulatory package has been developed. And we are now focusing on phase 3. So just the clinical phase for this drug so far has taken 9 years, and we still have a way to go with our phase 3 clinical trial. That is in a different population to the phase 2 study. It will provide both safety data and efficacy data in invasive aspergillosis in patients for whom an azole is not an ideal agent. So a different population, both to phase 2 and to what we've seen in the other invasive aspergillosis studies. So what is Olorofim? Well, it's a new oral agent. It's the first in class of the erotomides. It's a potent selective inhibitor of fungal dihydroorotate dehydrogenase type 2. And this is an enzyme that sits in the mitochondria and is involved in pyrimidine biosynthesis. And by impacting pyrimidine biosynthesis, it actually impacts multiple cellular functions, including cell cycle, DNA, RNA, and the cell structure itself. The spectrum is limited to molds. So DHODH is very different in yeasts and in humans. And this is a mold active agent. It is fungicidal, so it attacks and kills molds in multiple ways. And if you look at the photographs, in the top photograph, you can see a picture of an untreated Aspergillus hypha. You can see the nucleus there. You can see the cell wall is intact. And when you dose this with Olorofim, you get cellular rupture, you get breakdown of the cell wall integrity, and you can see that the nucleus is sitting outside of the cell structure. In terms of its microbiological activity, Olorofim actually has activity against a number of ultra-rare molds. So it's effective against Aspergillus, and we've tested more than 6,000 Aspergillus isolates, including cryptic and azole-resistant species. We've now tested over 300 Scedosporium isolates and over 100 isolates of Lamentospora prolificans, as well as Scopulariopsis and other rare molds. And using both standard CLSI and UCAST methodology, we see a fairly normal distribution of the MICs. So the blue bar shows you the MICs for Aspergillus, the green is Scedosporium, and the purple is Lamentospora. And whilst cutoff values have not been set, we are able to predict that if the MIC is less than 0.5 micrograms per mil, then it's highly likely that the mold will be susceptible to Olorofim. And the homology of the DHODH enzyme structure to that of Aspergillus is really key in predicting susceptibility. So if the homology of the enzyme is 50% or more that of Aspergillus, again, it's highly likely that the mold will be susceptible. We've done a number of in vitro and in vivo animal models. So the animal models have been done in Aspergillus, in Lamentospora, in Scedosporium, and coccidioides infections. On the right, you can see a neutropenic mouse model of inhalational invasive aspergillosis. And you can see that this is azole-resistant Aspergillus. You can see Olorofim in the green bar and posaconazole in the blue bar, with posaconazole being very close to the control outcome. It has good tissue penetration, including brain, bone, soft tissues, and lung, and levels in those tissues are at least the levels that we see in plasma. I'm just going to show you a couple of case examples from our phase 2 clinical trial. So this is a series of MRI images from a 19-year-old woman who had ALL. She was diagnosed with azole-resistant Aspergillus in both lung and CNS, and she had a biopsy-proven infection with the typical TR34L98H mutation identified by PCR, and this was whilst she was on chemotherapy for ALL. She had 8 months of high-dose posaconazole with intravenous and intrathecal amphotericin B, and she had some improvement of her lesions, but they did not resolve. When she stopped her chemotherapy, her antifungals were also stopped, and she presented following a seizure and with a focal periparesis. MRI scanning showed that one lesion had grown larger whilst the others were unchanged. So she was restarted on posaconazole and liposomal amphotericin B for another 4 months, but she did not have improvement of the lesions. She was enrolled in our phase 2 trial, and the lesions gradually resolved. She was on Olorofim for 14.5 months, but actually had complete resolution after that time and was able to return to university. And in the images you can see on the left at her initial diagnosis, you can see the ring-enhancing lesions. In the middle set of images, going top to bottom, you can see her images 3 weeks before she started Olorofim, and then the images on the right were after a year on treatment with Olorofim. And this is another interesting case. This was the first case we treated of a patient with Scopulariopsis infection. This was a female patient who had a single lung transplant, and she developed multi-drug resistant Scopulariopsis infection at the site of the anastomosis. She actually drove herself about 1,000 miles across the United States to enroll at a clinical trial site. She had 5 weeks of combination therapy with posaconazole, caspofungin, and terbinafine, and she was having very regular endobronchial argon plasma ablation, and she had a stent placed as well. In the image at the top, you can see at the 9 o'clock position, there's a little black area. That was the airway that she had left following laser ablation. So all of the matter on the right is burns material, scarring, and infection from this scopularis infection. The second image is after 10 days on Olorofim, where you can see she has a patent airway and some residual scarring, but otherwise, you know, a very good outcome. And then the bottom image is at day 22, where you can see almost complete resolution of her infection. So she was declared a complete success at day 84 and exited the clinical trial. And 11 months on, when she came back for follow-up, she had no recurrence of infection. So as part of the development process, you can obtain different regulatory designations, and these bring with them benefits in terms of both the pathway that you can take for approval for your drug and also potential extension on your patent life. So Olorofim has orphan drug designation in both Europe and the USA for Aspergillus, for Scedosporium infection, and a number of other infections. In the US, it has qualified infectious diseases product designation and priority review, which allow theoretically for a faster review process. And it's also the first antifungal to have been granted breakthrough therapy designation. So this was based on initial clinical data obtained from our phase 2 study. We went to FDA with some of the initial cases, and you can see there the statements. So invasive mold infections in patients with limited or no treatment options, a number of pathogens identified, and also treatment of central nervous system coccidioidomycosis, which is obviously not a big problem in Europe, but can occur in the traveling patient. And breakthrough therapy designation is an FDA process that is meant to support expedited development and review. It's for drugs that are to treat a serious or life-threatening condition, and where preliminary clinical evidence suggests that the drug may have advantages over existing therapies. So as a result of the breakthrough therapy designation, we took the rather ambitious step of submitting an initial filing in 2022 with the FDA with data from the first 100 patients. We have subsequently received a complete response letter from FDA just this month, requesting additional data and analyses from our study, and we've now closed enrollment with 203 patients in total. So what are some of the lessons learned? Well, antifungal development is really very slow, and new antifungal agents will be few. Truly novel agents will be even fewer, and they will take years to develop. So if Olorofim is approved on phase 2 data, it will be approved around 25 years since the company was founded, and about 20 years from the initial hit on the erotamides to having a clinical agent approved. And to do this development, you do need a streamlined clinical development plan, and you do need regulatory flexibility. You have to engage with the regulatory authorities in terms of scientific advice and getting help with your development program. And particularly for mold active agents, because these are less common than agents for Candida, stepwise development is important. You need initial studies, probably in very rare populations, in order to clearly identify an unmet medical need. And development can be facilitated by a focus on limited and well-defined populations where the benefit risk of a novel agent is appropriate. You're all very familiar with the agents already available. You know where they work, and you know where they're less likely to work or where there's likely to be an issue, and you choose your drug according to your patient profile. So there is a need to be flexible and pragmatic in terms of the benefit risk assessment, depending on that target population. And an unmet medical need is required to support development and approval where there is a more challenging benefit risk profile. Things like the limited population antibiotic and antifungal drug pathway in the US, conditional marketing authorization in Europe. These are very important pathways to try and bring these drugs to market in an earlier time frame than a traditional development program would allow. That's it from me. Thank you very much for your attention.
Thank you very much, Emma. Yeah, perhaps Martin, you are a specialist of new antifungal drugs, so please start.
Yeah. Thank you so much. So we have a question from Esther Segal from the audience.
A very interesting presentation, really. And one can say one should be crazy to go into that business. I am not a competitor, so I can say that. My question is, what do you do if by the time you eventually are on the spot where you can market, and your patent runs out?
Patent life is always a challenge. You don't necessarily get a patent very, very early in the drug development program. So there's a very fine art, I would say, between when do you seek a patent in order to maximize the patent life. Some of these development pathways, orphan drug designation, for example, can give you 6 months extension on your patent. If you do a pediatric development program, that can give you a 6-month extension on your patent. The reality is that for most companies, you have a very short window in order to recoup your investment after the drug is launched. So it's a bit of an art in terms of how you navigate that time frame. But the longer you take to develop your drug, then the shorter your patent life after you have your drug approved. We are fairly comfortable at the moment with the patent that we have on Olorofim and the time frame that we have to work with.
Perfect. Do we have more questions? Otherwise, I will jump in the meantime. I think we talked quite a bit today about cocci as well, just because, we have the fungal exposome and, climate change and all these areas where cocci plays a role, obviously, although, of course, you're right, there's not so many cases in Europe. There's mostly imported cases, although not so few, I guess, I think in Spain and in areas where there's more migration, basically, between countries where cocci is endemic and, where same language is spoken about, etc. So, but I think I just wanted to, ask you about official update, basically about what's presented, because I think, there's a number of very promising and breakthrough activities that we found, basically, for some of the fungal pathogens that are involved. Not saying that the results are not looking good for the others, but for cocci specific, I kind of mentioned before that, cocci meningitis is a lifelong that requires lifelong treatment. There's no cure. And there have been some indications, maybe, and you can mention a bit from what has been presented at ECMID or IDSA, that this may change, at least for some patients, with Olorofim. Can you a little bit elaborate on that?
Yes, certainly. So I actually took the cocci slide out of my presentation because this was predominantly a European meeting, but we have done in vivo animal models of a CNS model of cocci infection. And what you see with fluconazole, which is really the baseline therapy for coccidioidomycosis, is that fluconazole will suppress cocci infection. And when you then withdraw the fluconazole, you get rebound of the cocci in the CNS. What we found, though, with Olorofim, we tested 2 different doses of Olorofim. The mice were dosed for 2 weeks, and then they had 2 weeks off drug. What we found was that with the low-dose Olorofim, that suppressed the cocci in the CNS, but when you withdrew the Olorofim, you got rebound. But with the high-dose Olorofim, it suppressed the cocci infection, and when you withdrew the Olorofim, there was little evidence for rebound. So it appears that there may be a sterilizing effect on the CNS, at least in the mouse model. So our data to date in COXI, we've had 11 patients who completed the study and where those data have been presented. We have just completed, we literally just obtained all of the data from the second cohort of patients. We have a total of 41 patients with COXI who've now been treated in this study. I would say the signal is that the data are very comparable across the two groups. So we are hopeful that Olorofim is a drug that, if not life-saving in COXI, can be life-changing in COXI. We are hoping, and I think Martin knows this, to be presenting those data at the TIM Congress later this year. So I would say if you want to see the data, come to TIM, and hopefully you will see the data at TIM. But we have promising data. We hear very good feedback from the COXI clinicians on the symptomatic response that their patients have obtained with Olorofim particularly. We've had a number of patients who've had a Maya shunts who are receiving intrathecal drug, and patients who've had ongoing neurological deficit as a result of their CNS COXI. So this is a very sick population, and we've had patients respond extremely well. There was a very nice article in Scientific American last year where actually the first patient treated told his story, and he had CNS disease, but also very serious pulmonary disease. And at the age of 42 was oxygen dependent and mobilizing with a walking frame. So very disabled by his disease. He was started on Olorofim, got very rapid symptomatic improvement. And he has been on drug 4 years, and I think he's now elected to stop. He has some fixed neurological deficit, and he has fibrosis of his lungs, but he is able to mobilize normally, care for himself, go to the gym. He's no longer oxygen dependent, no longer dependent on a walking frame. So, that's a single case example, but certainly in COXI, that's the sort of impact that we think Olorofim can have for many of these patients.
Thanks so much, Emma. So this would basically the perfect word to go into dinner, but I think there's some other urgent question. Thanks so much also for referring to Tim. And actually, just from being one of the investigators in the study, it was really exciting also being part of this basically, because the focus started with Filomentospora and this data focus, but then this big emphasis came into basically Olorofim for COXI, and everybody was really keen in enrolling these patients for whom we really don't have any other options. And this continued, obviously, as you mentioned, the numbers in the second 100 patients, where really there was a bigger proportion even than in the first 100 patients of COXI patients. So very excited and looking forward to see the data. Tim, I think there is a question.
Yes. Last question?
Yeah.
Hello, thank you for your presentation. We know it could be very expensive to treat invasive fungal infections. So my question is, how much would it be to treat patients with Olorofim compared to other antifungal agents?
That's a very good question, which I can't answer at this point in time because we haven't actually decided on what the price should be. I think the reality is the price is going to be, many antifungals now are generic, and you have lots of generic options. This is a different type of drug and a newer drug, so it is going to cost more than many of the other antifungals, but I can't tell you at this point what that price is going to be.
Thank you so much. I think this is a perfect final word. Also, if we can kind of like have a curative treatment versus a lifelong therapy, this makes a big difference, obviously, if, data holds in that direction. And, definitely, thanks so much for doing all this.
Thank you.
You're very welcome.
And thank you, for presenting and summarizing the whole journey for us.
My pleasure.
Thank you, Emma. And it is very exciting to see that we have new families, but also all families with new drugs, such as risafongin, such as opelconazole. So in the next few years, it will be really interesting to see how all these drugs work. So I want to thank all the speakers now, and I remind you that you are all invited for the dinner at 8 o'clock at the second floor of the hotel, where we have where we had the lunch this at midday. So take a rest and see you in a few moments. Thank you.
Thank you.
Bye.